Hello everyone. Today we are talking about genetics. What is actually genetics? Generally, it is the science of study of inheritance. Inheritance से क्या मतलब? Something that is passed down from one generation to the other. Okay? That is called inheritance. So by definition, genetics is a study of science that deals with the structure, organization, transmission and function of genes and the origin of variation. What do we say? What is genetics? It is the science of inheritance and variation. The science of genetics deals with the principles that explain the similarities between the parents and their progeny and the difference among individuals of a single species. Now, why should a human being give birth to a human being and not to any other human being organism? So, generally, children resemble their parents. Is this due to hereditary? And they also differ from their parents to some extent because of variation. Now, heredity is defined as the potential of an individual to transmit its character to the offspring, while variation are the differences that the individual acquires due to interaction with the environment. The main aim of genetics is the study of heredity and variation. While heredity tries to maintain uniformity, variation brings plasticity. So, the term genetics was first coined by Bateson in 1906. History of genetics. Okay, there are three types, four main. So, the first is before Mendel, that is pre Mendelian era, between Mendel discusses Mendelian era, post Mendelian era, and modern concepts. History of genetics. People have known about inheritance for a very long time. Like, how do we know it? Because children resemble their parents, domestication of animals and plants, selective breeding of for good characteristics, Sumerian horse breeding records, Egyptian date palm breeding, ability to identify a person as a member of a particular family of certain physical traits. What is pre-Mendel? Pre-Mendelian area in which we study about the spontaneous generation, about telesocial, and about the theories of Louis Pasteur. So, what is spontaneous generation? Many biologists believe that some primitive organisms originate from non-living matter. What is non-living matter? That are decaying organic matter. Such origin of living organisms from non-living material is called as spontaneous generation. Now, Aristotle, who was the greatest biologist of early days, believed that not only plants but animals like fleas, mosquitoes, snails originated from decaying organic matter. But later, Lewis proved that organic matters decay due to the presence of microbes in them and that microbes do not originate spontaneous from the organic matter. Now, Louis Pasteur is known as the father of microbiology, which was disproved by Redi and Spallanzani but couldn't present experimental result. Now, this was Spallanzani experiment in which gravy is boiled and then the one of the flask is open, another is sealed. Gravy, which is open, it is terminating with microorganisms, and which is closed, the flask which is closed is free of microorganisms. Now, Louis Pasteur finally resolved it in an experiment, which is this one flask experiment that we already know, in which nutrient broth is heated. Over 1 year later, nutrient broth is free of microorganisms, curved neck is removed, and it is teeming with microorganisms. Reproduction in plants and animals. Babylonians and Assyrians in 700 BC. Now, Camerarius, however, who was the first time described sexual reproduction in plants, identifies sex organs in plants. In 1717, first artificial hybridization was produced by Thomas Fairchild, wherein the crossed Sweet Williams and Carnation and named it Fairy Child Mule. Joseph Corduter developed F1 hybrids in Nicotiana and Dianthus. Pre-formation theory. The homunculus theory of some of them suggested that the development of an organism is a simple enlargement of a minute but completely preformed individual. The preformed individual or homunculus could be either present in the sperm or in the ovum. Epigeneous theory, given by Wolff, opposed to the preformation concept, opinion that neither the egg nor the sperm had a preformed organism, but both of them have undifferentiated mass of living substance which developed into the organisms after fertilization. According to this theory, epigenesis, new organs and tissues are formed de novo due to vital forces. Transformation theory, it was proposed by Van Baer. He explained that the organs or tissues are developed by gradual modification of the embryo. Inheritance of acquired character given by Lamarck. He said that the characters are acquired in one generation is passed to the next. This is called the Lamarck's pangenesis theory. The pangenesis theory was proposed by Charles Darwin, in which he said that all parts of the body produce invisible gemmules or pangenes. He called the sex organs as pangenes, and these pangenes are assembled into gametes. And during fertilization, pangenes of mother and father combine to produce the character of offspring. This is also called the theory of evolution, in which he said the survival of the fittest is the reason that was also universally accepted. According to him, each gamete get one gemmule from one organ. This was his book of Charles Darwin, which is very important. This origin of species. Next is germplasm theory of Weizmann. Now, August Weizmann, he disproved the pangene theory with his experiments on rats. What he did was he cut the tails of rats and observed the progeny for 22 generations. According to his theory, the body of organism can be divided into two: somatoplasm and germplasm. Somatoplasm constitutes vegetative of the organism and germplasm is the reproductive part. Now, somatoplasm develops newly in every individual and there is no continuity, while germplasm, you can say it is continuous and links the generation. This is wide belief among the biologists during and before Mendel was the concept of blending inheritance. They thought that the medium of inheritance was liquid, possibly blood. However, this concept did not explain the experience of variation in F2. Now, this is Mendel's view of gene, which is or factor, as he said. They are particulate in nature, segregation during gamete formation, two different genes showing independent assortment or independent segregation. He used to refer the unit of inheritance, that is we call as genes, as factors. They were laws of Mendel, and he was Gregor Johann Mendel was the father of genetics. After 34 years, his finding was rediscovered independently by three scientists: Karl Lorenz, Hugo de Vries, and Schermak. Modern theory of evolution. Hugo de Vries was a botanist and geneticist. He recognized that organisms of different species still share common phenotypes, concluded that there must be a common gene in varied species. In the early 1900s, through observation of new forms of flower species, he introduced the concept of mutation. Bateson rediscovered and popularized the work of Mendel and proposed the term genetics. Bateson gave the term genetics. Then we have Johnson proposed the term gene. These are important. Bateson gave the term genetics and Johnson proposed the term gene, phenotype, and genotype. Walter Sutton and Theodore Boveri, they independently proposed the chromosome theory of heredity. Then there we have Yule, who proposed the idea of multiple factor hypothesis in 1906. 1908, Nielsen Helle explained the multiple factor hypothesis for grain color of wheat. Hardy-Weinberg law was given in 1908-1909, and Schultz gave the term heterosis in 1940. Classical genetics. is same. We know that gene genotype phenotype are also called pure line by Johnson. Genetics term was given by Bateson. Multiple factor hypothesis, Yule, Nilsson and L experimented. Morgan gave white eye gene in Drosophila, this sex linkage and X-ray mutagen, Mueller, Statler. Cell theory. Everyone knows this is a Robert Hooke cut a thin slice of cork and honeycomb chamber and he named that as cell. Scladen and Swan proposed different theories, like plant theory was given by Scladen, Swan animal cell theory. They saw it under the microscope. Now Nageli provided logical extension of cell theory. Some important discoveries: nucleolus by Robert Brown in Cell of Tradescantia, nucleic acid, Michener, mitosis, Fleming, meiosis, Farmer and Moore, chromosomal theory of heredity, Walter Sutton. Frederick Griffith. He was an English bacteriologist. He conducted experiments suggesting the bacteria are capable of transferring genetic information and such transformation are heritable. We have molecular genetics. We have Avery, MacLeod, McCarthy proved that DNA is the genetic material. Now, Alfred Hershey and Martha Chase in 1952 published a report confirming DNA as a genetic material. Biochemical genetics, Gerard in Born Error of Metabolism, and Beadle and Tatum proposed one gene, one enzyme theory. Watson and Craig gave the DNA double helix model in 1953. This discovery led to the new branch called molecular genetics. Barbara McClintock discovered jumping genes, or we call as transposons. Jacob and Monat lack operant concept. Benzer classifies cistron, recon, muon. Okowa in vitro synthesis of RNA. Kornberg in vitro synthesis of DNA. Okowa and Kornberg jointly were awarded Nobel Prize in 1959. Nirenberg and co-worker discovered the triplet genetic code. Some biotechnology aspect in genetic study. Carl Kreke is the father of biotechnology. He coined the term in 1919. The exploitation of biological process for industry and other purposes, especially the genetic manipulation of microorganisms for the production of antibiotic, hormones, etc. Genetic engineering, father Paul Berg. It is also called the genetic modification. It is a direct manipulation of an organism's genome using biotechnology. DNA fingerprinting was invented by Sir Alec John Jeffrey. In this technique, he used to identify individuals by characteristics of their DNA. PCR we know as polymerase chain reaction. It is a technique used in molecular biology to amplify a single copy or few copies of piece of DNA across several orders of magnitude, generating thousands of millions of copies of particular DNA sequence. You can see it. It was given by Carl D. Bank. Now this is the timeline that we will discuss in some other video. Thank you so much.